Do you like science, but you're not a practicing scientist? Have an idea you think is wonderful, but wonder how to find out? You could just give up and watch TV with a beer. But I had an idea and found a publicly available database from the National Institutes of Health that would give me the information I needed. Here's what happened. Do you ever wonder about all the life we see all around us? Plants, animals, bugs, bacteria? I wondered, is there something basic that all life has that isn't in things that aren't living? What do we mean by life? Different people have different answers, and Wikipedia says there is currently no consensus regarding the definition of life. But biologists have classified living things into the hierarchy you see here. Well, if we're looking for something found everywhere, we'd better start at the top. It turns out living things are in one of 4 domains: Archaea, bacteria, Eukaryota, and viruses. What makes them different from each other? Archaea were originally classified as bacteria. These single-celled organisms can live in extreme environments such as hydrothermal vents at well over 100 degrees centigrade, in alkaline or acid waters, in methane, in deep petroleum deposits, in your digestive system, and so forth. They're also abundant in the plankton of the open sea. Like bacteria, their DNA isn't enclosed in a nucleus. However, their cell walls contain different kinds of amino acids and sugars than those found in bacteria, and drugs that affect bacteria have no effect on Archaea. Bacteria are single-cell organisms with an estimated 5 × 10 to the 30th individuals and are easily the most abundant and diverse living things on Earth. They have no nucleus. There are about 10 times as many bacterial cells in your body as there are human cells, mostly on your skin, genital areas, mouth, and intestines. They're the germs that make you sick, but they also make vitamins, break down garbage, and maintain our atmosphere. Kill them at your peril. For nearly everyone, Eukaryota are the only living things they've ever seen. This domain includes all the plants and animals: mammals, birds, fish, insects, trees, algae, etc. The distinguishing feature is a membrane-bound nucleus that contains and protects the genetic material. Eukaryotes were the last to be formed and represent a tiny minority of living things. Viruses are small bundles of genetic material that don't have cells but are protected by coats of protein. They're generally not considered living things because they need to infect host cells to be activated and to reproduce. But they do have genes, which are molecules that cause things to happen, and evolve by natural selection. Their status became more controversial with the discovery of Pandora viruses in 2013 that are bigger than many bacteria, and, according to articles in Science magazine, their position in the tree of life is unclear. Viruses infect all forms of life. Individual living things are grouped at the lowest level as species. All living things contain cells, and all cells have DNA. What's that? Deoxyribonucleic acid, that's DNA, contains our genetic information, controls everything we do, and reproduces itself. It's the basis for all life. It's in the form of a double helix, and each one is called a chromosome. Each species has their own number of chromosomes. Almost all bacteria have 1 pair, wine grapes have 19 pairs, and humans have 23 pairs for a total of 46 chromosomes. Don't get too pompous because of the number. Chickens have 31 pairs. DNA has been known since 1869 and its importance in genetics since 1927, but its actual structure as a double helix and how it works wasn't recognized until 1953. Watson, Crick, and Wilkins won a Nobel Prize in 1962 for that description. Franklin did the critical X-ray work, but died in 1958, and the Nobel folks like to award the prize to living people. Gosling was a grad student who helped, and as you know, students aren't considered living until they graduate. Okay, here's DNA again. The two silver bands forming the double helix are a sugar phosphate backbone. In this case, sugar is good for you. Inside the helix are chemical nucleic acids called base pairs. Chromosomes could have a lot of base pairs. Humans, for example, have over 3 billion. Don't get a swelled head. The short-tailed opossum has more, and so does the chimp. There are 4 nucleic acids: adenine, thymine, guanine, and cytosine. Biochemists refer to these nucleotides by their first letters, A, T, C, and G. Because of how their chemical bonds hook up, adenine matches with thymine, and guanine matches with cytosine. It's not perfect, but those are the best matches. So, the DNA base pairs is the place to look for something in common. But the string has to be long enough that it can't be by chance. How long is long enough? It seems to me that if something happens by chance only once in a billion, that's good enough. In this case, with 4 possible nucleic acids, that's 15 base pairs in a row that has to be in every species to find something in common. Matched sequences should be ignored if they're just filler or repeated many times. What's interesting enough to consider? To quote U.S. Supreme Court Justice Potter Stewart when he was describing pornography, "I know it when I see it." The National Institutes of Health, NIH, maintains an online database at the National Center for Biotechnology Information, that's NCBI, with complete DNA sequences for a variety of species. I was able to check 30 Archaea, 66 bacteria, 48 Eukaryota, and 19 virus species. You'll see my data towards the end of this video, but for now, follow what happened. If a match was to be found everywhere, it also had to be in a species with the smallest number of nucleotides. In this case, it was 2 bacteria, Carcinella rudi and Mycoplasma gentialium. A 15 nucleotide match was found in these 2 bacteria. Note the symmetry, beginning with CGGGTT and ending with TTCCCG. Further inspection found it in other bacteria and Eukaryota. Things were looking good. Things were looking better. All 48 species of Eukaryota had it. But not all the bacteria species had it. And it wasn't in 2 species of Archaea. But there was still hope, because the NCBI database only displays one of the 2 strands of the DNA double helix. The other strand is its reverse complement. Reverse complement? What the heck is that? To get the reverse complement, start at the end, which is the reverse, of the sequence and change each nucleotide to the one it connects to, its complement. C connects to G and A connects to T. Looking at the found sequence, the end nucleotide is a G, so we change it to a C and write it at the beginning. Next are 3 C's, which become 3 G's, and so on. Nope, it wasn't found everywhere. Apparently there is no interesting and unique sequence in the DNA of all living things. To ease my sadness, I had another glass of wine. Yes, drinking a depressant when you're depressed is depressing, but for me just a little helps to clear the mind. If Eukaryota has a special sequence, I wondered if bacteria and Archaea have ones of their own. I picked a couple of small Archaea species and found this 17 nucleotide sequence that could occur at random only once in 17 billion times. But bacteria weren't cooperating at the 15 nucleotide level, So I lowered my standards and found a 13 nucleotide sequence that was in all of them, occurring at random once in 67 million times. So here they are, very rare sequences that are specific to each domain. Are they part of something the body uses? Yes, they are. They're part of a molecule called transfer ribonucleic acid, usually called tRNA. What's it used for? Here's a drawing that I'll explain in a minute. There's a lot of amazing stuff going on in each cell of your body, and this is a little of it. tRNA helps the body make proteins from amino acids. The developing protein is the pink chain. A complex mass called a ribosome moves along a chain called messenger RNA, that's mRNA, reads every 3 nucleotides, attracts a tRNA with the matching nucleotides, attaches the amino acid on the tRNA to the protein chain, and releases the tRNA to find more. If you look closely, you see the incoming tRNA has AAG that matches with UUC in the mRNA. The nucleotide uracil, which is a U, takes the place of thiamine, a T. So here's what tRNA looks like as a simple drawing. The part for our sequences is labeled T with that little symbol C. That little symbol is pseudouridine, which is a chemical that is part of that. And it's called that T symbol C loop, also called the T-arm. Drawings are two-dimensional, but our cells have 3-dimension, and tRNA looks a bit more complicated in real life. Pretty messy, huh? Complicated, too. The colors correspond to the drawing. Okay, here are our sequences arranged as the T-arm is. Now things begin to make sense, with the sequences on either end matching up with each other in a flower-type arrangement. By the way, there are a lot of different sequences for the T-arm, not just this one. And there are the flowers without the stems. If tRNA just attaches amino acids to protein chains, why is the T-arm different for each domain? Let's look at the data. The first column is the domain, the next is the species' scientific name, and the third is the number of nucleotides in it. Sequence 1 is the one for Archaea, sequence 2 is the one for bacteria, and sequence 3 is the one for Eukaryota. Notice that the only link of sequence 1 with sequence 3 is for something in hydrothermal vents, hot oil deposits, and hot springs. And sequence 2, which is bacteria, is pretty much on its own except for botulism, tetanus, and diarrhea in Archaea and Eukaryota. And sequence 3, which is Eukaryota, has a lot of matches with sequence 1, which is Archaea, except for fungi, algae, protozoa, and nematodes. Also, that none of the sequences are in any virus. What might this mean? Not all species of bacteria have T-loop stems starting with CC and ending with GG. Some have a variety of nucleotide pairs, so X's have been used to fill them out. Now the question is, could the data suggest if a bacteria or an archaea was the first living organism? Let's look at the nucleotide differences for each domain. Well, let's look at scenario 1, that Archaea came first. The mutation of a single nucleotide in Archaea could form the bacteria sequence. However, for Archaea to create Eukaryota, 2 nucleotide mutations would be needed. So the tentative indication is Archaea did not come first. Well, here's scenario 2. Bacteria came first. The mutation of a single nucleotide in a bacteria T-loop could change it to form the Archaea sequence. While Archaea are well known in extreme environments, they're also plentiful in ocean plankton. Perhaps another single nucleotide mutation occurred in a bacteria that was in an archaea to form the Eukaryota sequence. This scenario could also explain the presence of Archaea sequence nearly throughout the Eukaryota domain, the near absence of the Eukaryota sequence in Archaea, and the near absence of the Archaea sequence in bacteria. Also, another single mutation in an organism with a bacteria that wasn't in an archaea could explain a fungi, algae, protozoa, and nematode sequence exception. I realize that all this sounds complicated, so have another glass of wine. Our tentative indication is bacteria came first. So in summary, the original goal of this research, to find the same DNA nucleotide sequences in every species that could not occur by chance, was unsuccessful. There are no sequences that are at least 15 nucleotides long that occur in every living thing. However, a common sequence for each domain was tentatively identified. All of these sequences comprise the DNA template for the tRNA T-arm, and none of them are in any virus. For this project, 163 species, 30 Archaea, 66 bacteria, and 48 Eukaryota and 19 DNA-based viruses were thoroughly inspected. It was noticed that the Archaea sequence occurs in all Eukaryota except fungi, algae, protozoa, and nematodes. This leads to a suggestion that those species originally came from an organism with the bacteria sequence mutation, but without the Archaea sequence, while all the other Eukaryota originated from an organism with the bacteria sequence mutation that included the Archaea sequence. As I'm sure you realize, there are still questions to be answered. Here are a couple of them. Why are these particular sequences domain-specific? Why are the sequences in the T-arm of tRNA? Does tRNA do more than assist in protein creation? That's the story of this independent research. But let's chat for a minute on independent research in general. Are you a free thinker with a novel and exciting idea that you want to develop? If it's a science idea, you'll find lots of information and data available, but you have to look for it. You should also realize that you'll be spending an awful lot of time alone researching your idea, with an awful lot of ideas being wrong, perhaps including my idea you just saw. And you're not getting paid. You have to really believe in your idea and in yourself to have any chance of success. If you do find something, you can certainly be proud of increasing world knowledge, but be careful how you express your findings. Try to use words like perhaps or speculate rather than prove or known, and have data around to back you up. Keep in mind that even Darwin's theory of evolution and Einstein's theory of general relativity are still called theories and not facts. It also helps to have someone who inspires you. In my case, it's my daughter Heidi, who was stricken with a brainstem tumor, but who continued helping troubled teenagers while becoming progressively paralyzed. Here's what she told CBS News.